guys, welcome to my YouTube channel. I'm Dr. Shuchita Verma and today I'm back with another video on biotechnology. So let's get started. So in our last video, we had an introduction to biotechnology, that what is biotechnology and what are the various applications and a little historical background about the biotechnology. So to start with this, I'm going to first take up the topic for what are the enzymes that are being involved in this biotechnological processes. Yeah, so let's start with enzymes. Now there are different categories of enzymes that are very useful in the biotechnology. The very first enzyme that I'm going to discuss here are the lyases or the lysing enzymes. Lysing enzymes. So lysis means a process that includes the rupture of the cell membrane or to break down. So whenever there is a word lysis or lysis, then it means to break down, that we are breaking some component into smaller pieces. Yeah. So lysing enzymes. Now what is the role of these enzymes in biotechnology? So the first thing is that we need a gene of interest to be inserted into the vector, yeah, where we want to get that gene transcribed and then ultimately translated into the protein or the product of our choice, of our interest. So the gene of interest, basically how do we get it? Yeah. So to get the gene of interest, we all know that this gene is basically present inside the nucleus. So to reach up to that nucleus, we need to break down the cell wall. So it is present inside the nucleus of the cell. We need to break down the other layers of the cell so as to reach the gene. Now, here comes the role of these lysing enzymes and how we do that. We'll take an example of a plant cell. So we all know that a plant cell has a cell wall. So the very first layer is a cell wall, and this cell wall is actually made up of cellulose. Yeah. So to break down this cellulose, we need to have the enzyme cellulase. So this is the lysing enzyme that is used to break down the cellulosic cell wall of a plant cell. Then apart from cellulose, there could be other things, other substances present. Supposedly pectin. Pectin is present. So there is another enzyme known as pectinase, which is helpful for the breakdown of pectin. Yeah. This is about cell wall. Coming on to the next layer is the cell membrane. The cell membrane is again made up of phospholipids and various proteins. So to break down the proteins, we would need proteases. And for the breakdown of phospholipids, we need lipases. So this is in case of plant cell. Now let me consider the fact that we have to isolate from the animal cell. If I take an example of the animal cell, then the animal cell does not contain cell wall. So we don't need these two enzymes. Yeah. We don't need these two enzymes because the animal cell contains cell membrane, and cell membrane is then composed of phospholipids and proteins. So to digest the proteins, we need proteases, and to digest the phospholipids, we need lipases. Yeah. Then there could be another example. Supposedly, I need to isolate a gene from fungus. Yeah. So my cell is now a fungal cell. And in this case, the cell wall is made up of chitin. So the enzyme used to dissolve this chitin is chitidase. Alright. Then another layer is then again cell membrane. So we will need proteases and lipases. Proteases and lipases. This is about fungal cell. Now I want to isolate the gene from a bacteria. How do we do that? Now I want to isolate a gene from the bacterial cell. How do I go about it? So we know that bacterial cell wall is made up of peptidoglycan. Yeah. So, and we also know that there is an enzyme known as lysozyme which helps in the lysis of a bacterial cell. So instead of taking into account all other we are straight away going to use this lysozyme enzyme, this lysozyme enzyme for the breakdown of the bacterial cell. Yeah. So this is about the lysing enzyme. Next, we move on to the another category of enzyme that are endonucleases. So next is endonucleases. So endo means inside and nucleases means which break down the nucleic acid. So endo means inside and nucleases are the enzyme that breaks down the bonds between the nucleic acids inside the nucleic acids. Yeah. So now if I talk about endonucleases, these enzymes, they make two types of cuts which we are going to discuss in detail further. But before moving on to that, I would like to introduce here how the scientists got to utilize this concept of endonucleases for the biotechnological processes. Now what happens when there is a bacterial cell? Now this is a bacterial cell. Now this is a cell of E. coli, Escherichia coli. Yeah, and this is the genetic material of E. coli. So now E. coli is a prokaryote, and we know that in prokaryotes there is a defense system which is a two complex system. So there is a defense system that is a two complex system. The first system contains of the methylating agents, and the second system consists of endonucleases. Now what happens when a virus tries to invade? Now this is a virus. It tries to invade the bacterial cell and tries to transfer its DNA into the bacterial cell. Now what happens? The endonucleases and the methylating agent both act in coordination to restrict these viruses to take over the host cell or to take over the bacterial cell. We'll study this with the help of an example. Now consider that there is one pair A, T, and the same pair is here in the bacterial genetic material A, T and A, T. Yeah. So now what this endonuclease is going to do? The endonuclease has the ability to make a cut in between the base pairs. Yeah. So this endonuclease will make a cut between here A and T, and also it will make a cut between this A and T. Now consider the situation that this endonuclease is trying to save the E. coli cell by disrupting or disintegrating or breaking this viral DNA. But in that process, it is also able to recognize the AT bases here and then finally breaks down this also. So what happens? The virus DNA will be broken from here. Similarly, the bacterial DNA will also be broken from here and here. Now, this condition is a suicidal condition for the E. coli, which the E. coli does not want. To save this from the condition, there comes the role of the methylating agents. These methylating agents, they add a methyl group onto these bases of the bacterial cell, bacterial DNA. So now what happens is that when the endonuclease tries to break down the viral DNA, it will recognize this AT base pair, but it will not recognize these methylated ones. So it is only going to break the viral DNA and is not going to affect the bacterial DNA. Now, because the bacterial DNA is already methylated. So this is the defense system present in all the prokaryotes, and because of this defense system, we, the scientists, are now able to, find out a solution with the help of these endonucleases, that these endonucleases can be used to make cuts in between the DNA molecules. And by making the cuts, we are now able to insert our gene of interest for the transcription and translation of the desired protein. Now, when we talk about the endonucleases, we call these endonucleases as molecular scissors. We call them as molecular scissors, and the reason is that they make a cut in the DNA at a specific site. So for that reason, these endonucleases are called as molecular scissors and are a very important tool in our DNA technology, that is the recombinant DNA technology. So now the third category is alkaline phosphatase, and see what happens. See, this is our plasmid. Yeah, this is our plasmid. And we all know that plasmid is the extra chromosomal genetic material that is present in the cell. So now plasmid, we need to insert our gene of interest into this plasmid. So to insert our gene of interest, we need to break open this plasmid. So how am I going to make a cut? I will be adding an endonuclease, and this endonuclease is going to make a cut here. So now my plasmid will open and I will get this plasmid in a linear form. So now this is also a DNA but in the circular form, and this is also the DNA but in the linear form. So now this is the 3' end. At this 3' carbon, there is a hydroxyl group attached. And this is the 5' where a phosphate group is attached. So we know that to make a phosphodiester bond, we need phosphate as well as we need a hydroxyl group. Yeah. So whenever I am making a cut with the help of an endonuclease, my plasmid is getting open into a linear form. But because at 3' there is an OH and at 5' there is a phosphate, it has a tendency to make a circular form again, to join again, yeah, for forming the phosphodiester bond. Now again the situation comes that I introduce an endonuclease, it makes a cut, and then I get a linear DNA. But then because of the tendency to form a phosphodiester bond, again there are chances that they join again to form the circular form. Now I want to avoid this situation. Here comes the role of the enzyme alkaline phosphatase. So I will now introduce alkaline phosphatase, and what this enzyme is going to do, it is going to remove this phosphate from this 5' end. Now what am I left with? Once I remove this with the help of alkaline phosphatase enzyme, I am going to get 3' OH but only 5 prime here. So now this kind of DNA where there is hydroxyl present but then at 5 prime there is no phosphate present is unable to make a phosphodiester form, hence it will remain in this particular linear form. Now in this form I am able to add my gene of interest, yeah, and then because my gene is also a DNA, so it will also have a 3 prime carbon with a hydroxyl group and a 5 prime phosphate. Now this phosphate and this hydroxyl group will be able to make a phosphodiester bond. Yeah, and then when there is a phosphodiester bond, my gene of interest is now added to the plasmid. Alright. So now when there is a formation of this phosphodiester bond, what we are going to get? We are going to get this open plasmid and along with this insert. Yeah, we are going to get this open plasmid along with this insert. I'm just making it a little darker so as it is supposed to make it more visible. Yeah. So this is my open plasmid and this is my gene of interest or the insert that I have added. This is a linear form. Now to make it back to the circular form, I will be adding phosphorylases, which then adds the phosphorus at the 5' end, and then there could be a formation of a phosphodiester bond so as to make it a circular form. Yeah. Then another thing which is important, that how do I join these two DNAs, the open plasmid DNA and the insert. Now here comes the role of another type of enzymes that I'm going to discuss, and that is ligases. So basically the keyword here is to join. They're used to join different DNA segments. So whenever we have an open plasmid DNA and our gene of interest, so with the help of these enzymes, ligases, we are able to join the plasmid DNA with our insert. Yeah. Next category of enzymes are phosphorylases. These enzymes, they add phosphate groups. Yeah, they add phosphate groups so as to help the formation of the phosphodiester bonds. Alright, so these are the basically 5 types of enzymes that are used. First are the lysing enzymes that are used to break down the different layers like the cell wall or the cell membrane so as to reach to the nucleus. Then comes the restriction endonucleases which make a cut in the DNA at the specific sites. And why they are deemed as restriction? Because they are restricting the other DNA to take over the host cell. Yeah. Then third types are alkaline phosphatases, where where they are used to remove the phosphate group from the 5' end so that the plasmid DNA could be in the linear form and we will be able to add our gene of interest or the insert to it. Yeah. And fourth are ligases, which are used to make bonds between the plasmid DNA and our insert. And fifth are the alkaline phosphorylases, which adds phosphate groups so as to make the phosphodiester bond. So these are all about enzymes used in these processes. And as far as restriction endonuclease is concerned, I'm going to make a separate video where I'm going to tell you about the different cuts, the blunt cut, the staggered cut that these restriction endonucleases make, and how they make these cuts, which cut is more important, is more comfortable to work with, and what is the criteria for the nomenclature of these restriction endonucleases. If you have any query, then you can ask me in the comment section, or just let me know how do you like the video. Was it informative enough? What are the things that you want me to discuss in upcoming videos. And please do like and share the video, and don't forget to subscribe to the channel and press the bell icon so that you don't miss any further videos. Till then, goodbye.